So I'd like to start off by acknowledging my supervisors, members of my advisory committee, my lab members, as well as the following sources of funding. So mild traumatic brain injury, mTBI, commonly referred to as concussion, is known to be the leading cause of neurological disability in young adults worldwide. And in Canadian residents, there's around 200,000 cases being diagnosed annually. And two groups of people that are at an increased risk for mTBI are those involved in contact sports such as hockey, football, rugby, as well as military service members. So unlike more severe brain injuries called traumatic brain injuries, there is no large-scale neurological damage following an mTBI. And this is a big problem as most neuroimaging techniques probe relatively large spatial scales, being on the order of millimeters. They're unable to detect pathological changes that happen after a concussive impact. In particular, this makes the study of long-term consequences following repeated concussive impact very challenging. So diffusion MRI is a technique that makes the MR signal sensitive to the diffusion of water molecules within the tissue, and this allows us to probe tissue on a microstructural level, enabling detection of these changes that happen after an mTBI that are invisible to conventional neuroimaging techniques. The most common analysis technique for diffusion MRI is called diffusion tensor imaging, and this allows us to obtain metrics such as mean diffusivity, which gives a measure of water molecule diffusion averaged over all directions, as well as fractional anisotropy, which quantifies the directional preference of diffusion within the tissue. So for example, if we're looking at an axon, we would expect water molecules to be more likely to diffuse along the direction of the axon as opposed to into the membrane. So this would result in an increased FA value as compared to a cell body, which is approximately sphere-like. So despite DTI being a step forward in the study of mTBI, studies in rodent mTBI models report insignificant changes, and findings have not been consistent across studies. And this emphasizes the need for more sensitive and specific techniques. So for this study, we're using advanced diffusion MRI, which addresses two key limitations in conventional methods. So the first one is that diffusion tensor imaging assumes a Gaussian distribution of water molecule diffusion. And although this is accurate in the case of uniform or free water diffusion, where there's little to no barriers present to impede diffusion, in biological tissue, there's various cellular and subcellular structures that impede diffusion. And so in this case, the distribution is more accurately represented using an additional kurtosis term. So in this study, we'll be quantifying kurtosis, which is called diffusion kurtosis imaging. And this gives further insight and it is also more sensitive to changes in tissue microstructure as compared to conventional DTI. And secondly, conventional diffusion MRI uses what's called pulse gradients to encode diffusion, which is shown in the figure here. However, due to limitations in hardware, we are unable to probe smaller spatial scales being on the order of under 10 microns with this technique. So to address this, we're using oscillating gradients, which is shown which the waveform is shown here, in addition to these conventional pulse gradients. And this allows us to probe those spatial scales under 10 microns and also allows quantification of more advanced metrics, which I will discuss in coming slides. So the overall objective of this work is to characterize how advanced diffusion metrics change in the long term following repeated mTBI in a mouse model. So the data collected for this project are from transgenic mice, which contain humanized versions of both tau and amyloid precursor protein. In terms of the imaging sessions, there's an imaging session performed at baseline when the mice are around 6 months of age. And then a week after this session, mice in the mTBI cohort undergo an mTBI procedure on 3 consecutive days. And then another imaging session is performed 6 months after the injury to look at any changes. And we currently have data from 5 mice in both sham and concussed cohort with 3 males in each group. In terms of the imaging protocol, we acquired diffusion data at 3 different gradient oscillation frequencies. So the first is 0, so this would be a pulse gradient acquisition, and then also at 60 and 120 hertz. And these have corresponding effective diffusion times of 12, 4, and 2 milliseconds. And what effective diffusion time is that controls how long we let water molecules diffuse within the tissue before collecting the signal. And so, for an example, as we increase the gradient oscillation frequency, we decrease the effective diffusion time, and that means that we are probing a smaller spatial scale within the tissue. And at each of these frequencies, we acquire data at 2 B-value shells, each with 10 directions. And the total diffusion scan took around 66 minutes. And as part of this study, we also acquire an anatomical T2 weighted scan. In terms of how we actually calculate these metrics, diffusion tensor, so MD and FA, as well as mean axial and radial kurtosis, were acquired using the recently introduced axisymmetric DKI fitting, which was performed on the data from each frequently separately. We also implemented spatial regularization during the fitting process to reduce noise in subsequent parameter maps. In terms of these advanced metrics that I alluded to earlier, to explore how these various metrics change with diffusion time or with the spatial scale that we are probing, we can calculate diffusion time dependent metrics or delta metrics. So what these metrics are each of the metrics encoding using the oscillating gradients, so that would be the smaller effective diffusion time, minus the metric encoding using the longer effective diffusion time or the larger spatial scale. And the idea behind this is that these metrics are known to be more sensitive to the underlying microstructural changes as compared to metrics only acquired at a single diffusion time. We have currently looked at 6 different ROIs, being the corpus callosum, the internal capsule, and the fornix for white matter regions, and the entorhinal area, the cortex, as well as the hippocampus. And in terms of how we actually get mean values of these metrics, we get these metrics from a brain atlas, which we then register to the T2 weighted anatomical for each imaging session, and then down to the native diffusion space to acquire these mean values. And shown here is a set of representative parameter maps taken from one of the mice at baseline. So in the top row, this is data acquired using our pulse gradient acquisition. And we can see each of the 5 metrics being mean axial radial kurtosis, as well as the diffusion tensor metrics on the right-hand side. And shown in the bottom row is the more advanced time-dependent metrics of the metric shown above it. Now jumping into some results. So shown here is data from our pulse gradient acquisition. So that's the top row that I showed on the previous slide. And each of these separate graphs depicts one of the 5 metrics that we're looking at. And each of the regions of interest are along the x-axis. And what's actually shown on the graphs here is the average of the metric within each cohort at 6 months minus the baseline. So we're just looking at the change relative to the baseline value. In terms of statistics, we only detect a significant group effect indicated by the stars here, or the asterisk here, for mean diffusivity, and we don't detect any significance through follow-up post-hoc testing. However, we do see some consistent trends in these metrics between the groups, and that is a decrease in mean axial and radial kurtosis, as well as an increase in mean diffusivity in the concussed cohort relative to the sham cohort. And so this particular set of metric changes are consistent with changes such as loss of neurons, demyelination or damage to the myelin sheath, as well as damage to the axonal membrane. And so moving on to our more advanced time-dependent metrics, we have the same graph setup as in the previous slide. And what we can initially see is that we detect a lot more significant difference between the groups, detecting a significant group effect in 4 out of the 5 metrics that we're examining. And we also detect significance through follow-up post-hoc testing. So firstly, in terms of changes, we see a consistent trend of decreased delta mean kurtosis in the concussed cohort relative to the sham. And based off past studies, this would indicate loss of neurons in the gray matter, as well as demyelination in the white matter in the concussed cohort. And secondly, we see a significant decrease in delta axial kurtosis in the concussed cohort in the entorhinal area specifically. And as some of you may know, this is one of the first regions to degenerate in Alzheimer's pathology. So this may mean that this metric may be sensitive to early Alzheimer's pathology. And finally, we see large and significant differences in delta radial kurtosis, delta MD, as well as delta FA in the internal capsule. And this particular set of metric changes may suggest altered axonal packing, as well as damage to the myelin sheath following an mTBI. So the key takeaway from this study is that advanced diffusion metrics provide greater sensitivity as well as specificity following brain injury as compared to more conventional diffusion metrics. So in conclusion, while imaging markers of repeated mTBI remain challenging to detect, we here show sensitivity to long-term microstructural changes following repeated concussive impact using both conventional and advanced diffusion MRI. In terms of future directions, we are going to increase sample sizes as well as investigate data that is acquired 1 year post-injury to see if these metric changes persist or even worsen at that time point. We're also going to investigate various genotypes and how these interact with mTBI, as it may be possible that certain genotypes may be more predisposed to have a worse outcome following repeated concussive impact. And finally, the work I presented here is part of a much larger study involving multiple research groups using various techniques to study the long-term consequences of repeated head injury. And this allows us to correlate changes in these diffusion metrics with results from functional MRI, histology, as well as behavioral measurements. And this allows us to better understand what changes in these diffusion metrics represent in terms of both structural damage as well as cognitive impairment that occurs following brain injury. So with that, I'd like to thank you for your attention and welcome any questions.